Sorry for the delay. I think Shahab got some technical issues. He's here. So we will start again by the recitation of Holy Quran. Shahab, please join us.
کریں آواز آ رہی کہ نہیں؟
Yes, Shahab, you are audible.
اچھا آواز آ رہی ہے نا؟
Yes, you are. Yes, you are audible.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم هو الله الذي لا إله إلا هو عالم الغيب والشهادة هو الرحمن الرحيم هو الله الذي لا إله إلا هو الملك القدوس السلام المؤمن المهيمن العزيز الجبار المتكبر سبحان الله عما يشركون هو الله الخالق البارئ المصور له الأسماء الحسنى يسبح له ما في السماوات والأرض وهو العزيز الحكيم پناہ مانگتا ہوں میں اللہ کی شیطان مردود سے، شروع اللہ کے نام سے جو بڑا مہربان نہایت رحم والا ہے۔ وہی اللہ ہے جس کے سوا کوئی عبادت کے لائق نہیں، نہاں و عیاں کا جاننے والا، نہایت مہربان رحمت والا۔ وہی اللہ ہے جس کے سوا کوئی عبادت کے لائق نہیں، بادشاہ نہایت پاک، سلامتی دینے والا، امان بخشنے والا، حفاظت فرمانے والا، عزت والا، عظمت والا، تکبر والا، اور پاکی ہے اللہ تعالیٰ کو ان کے شرک سے۔ وہی اللہ ہے بنانے والا، پیدا کرنے والا، ہر ایک کو صورت دینے والا، اسی کے ہیں سب اچھے اچھے نام۔ پاکی بولتا ہے جو کچھ آسمانوں میں ہے اور زمینوں میں ہے، وہ عزت والا، حکمت والا ہے۔ صدق اللہ العظیم۔
Jazakallah. Thank you, Shahab, for these beautiful verses of Holy Quran. Now, we should start the proceedings, and now I will invite Dr. Humaira Yasmin, and she is requested for her welcome address. Before starting her address, let me give some introduction of Dr. Humaira Yasmin. Dr. Humaira Yasmin is an assistant professor of applied microbiology and plant physiology at the Department of Biosciences, Comsets University, Islamabad. And talking about her research interest, her research interest includes searching for economic, eco-friendly, and sustainable strategies for crop improvement under biotic and abiotic stresses. Her research work focuses on understanding the detailed biochemical mechanism adapted by crops when exposed to biotic pathogens and abiotic stresses. Abiotic stresses include drought, salinity, heavy metals, and heat. And she also works on green synthesis and characterization of nanoparticles from medicinal plants, application of growth regulators, chelators, and other chemical compounds to increase plant production and these processes are under investigation in her lab. Her group have developed methods to understand the mechanism of action of target molecules using bioinformatics expertise, including docking analysis, to estimate the binding of moieties of molecules to the target receptors, and expressional studies of potential stress related genes. She has been awarded with fellowships for PhD QAU from Qaidam University, Islamabad, Pakistan, IRSIP, University of Sydney, Australia, travel grant for ICP SR 2019, Baltimore, USA, and postdoc fellowship in USA also. And all of these awards were awarded from Higher Educational Commission of Pakistan. She also published almost 52 articles in national and international journals in the field of plant physiology, plant biochemistry, plant pathology, and plant microbe interaction with impact factor of almost 150. Moreover, she is teaching and supervising BS, MS, and PhD students with different research projects. She is an active member and core committee of PAC PGPR Society. She is also working as a principal investigator of a research project worth 5.9 million PKR for the development of novel biofertilizer strains. So I would like to invite Dr. Humaira Yasmin for her welcome address.
Thank you so much, Tayyib, for introducing me so well. Can you hear me properly?
Yes, ma'am, you are audible.
Okay. Honorable guest speakers, respected participants, and dear students, Assalamu alaikum and good afternoon. Welcome everyone, and thank you for joining us for today's webinar. We are pleased to be able to welcome those of you that have been with us for years now, as well as those of you who are new to this society. I'm pleased to welcome you to this webinar on World Soil Day with the theme of Keep Soil Alive and Boost Soil Productivity. Basically, World Soil Day is celebrated annually on December 5th as a means to focus attention on the importance of healthy soil and to educate for the sustainable management of soil resources. And as an international day to celebrate soil was recommended by International Union of Soil Sciences in 2002. As you know, the soil health is a major global issue, and moreover, our agricultural systems are also vulnerable to threats of climatic changes and environmental pollution. So it is the dire need of our to direct our research of finding solutions to the challenges faced by the plant and soil. And plant growth promoting Rhizobacteria, which are abbreviated as PGPR, they have been studied from decades, and they have shown an important role in the sustainable agriculture industry. The increasing demand for crop production and while a significant reduction of synthetic chemical fertilizers and pesticide use is a big challenge nowadays. The use of PGPR has been proven to be an environmentally sound way of increasing crop yields by facilitating plant growth through various direct and indirect mechanisms. We are highly thankful to our respectable speakers for sparing their precious time for this occasion. And our speakers include Professor Dr. Riaz Sayed, a professor and head of Department of Microbiology, PhD, VP, Mandal Arts and Science College, Shahada, Maharashtra, India. And the second speaker is Dr. Nasheen Ilyas. She is an assistant professor in the Department of Botany, Peer Meher Ali Shah Arid Agriculture University, Rawalpindi, Pakistan. And our third speaker is Dr. Muhammad Nadeem Hassan, assistant professor in ComScience Biosciences Department, working on the enumeration and identification and characterization of plant-associated rhizobacteria for biofertilizer, biopesticide, and high-value industrial products. I welcome all you in this webinar. And I also warmly welcome Professor Dr. Asghari Banu, our mentor, leader, and role model, to honor this occasion. I'm grateful to her, despite the time difference. She has been accepted my invitation generously and will present vote of thanks to all the guests and participants at the end of the webinar. I hope you all have great learning and experiences in this webinar. Our first speaker today is Professor Dr. Riaz Sayed. I'm not sure he has joined it yet or not, because he was having some problems. Dr. Riaz,
Dr. Umar,
have you joined?
I have joined.
Okay.
Yes,
Okay.
yeah, I have joined.
Okay. And let me give some introduction to our respected speaker, Professor Dr. Riaz Sayed. Currently, he serves as the president of Indian chapter of Asian PGPR Society for Sustainable Agriculture. The Asian PGPR Society has been established in 2019, and he has been actively coordinating the society international conferences in Asian countries over the years. To his credit, he has received the Best Teacher Award, Young Scientist Award in 2005, 2008, and in 2012. Professor M.M. Sharma Award, Springer Society Award in 2020, and he has also been awarded the excellent contribution to the Society of Environmental Sustainability, and also got award for excellence in PGPR research. He is associate editor of Environmental Sustainability, Springer Journal, and he has over 25 years experience of teaching and research, and 20 years of research experience in microbiology and biotechnology. His research expertise is a cytophore-based microplant interaction and microbial biopolymers. His publications have been widely cited in the field of cytophore-based PGPR. And he has also trained several graduate and postgraduate research students and supervised 6 PhD as well as authored and co-authored over 198 research papers in reputed national and international journals. He has also edited 25 books and 50 textbooks and 4 proceedings of the Asian PGPR International Conference. He has delivered many invited talks in several Southeast Asian and European countries. Today, he will deliver his lecture on Rhizobacteria, legendary guards and health promoters for soil and plant. Over to you, Dr. Riaz. You can start your lecture, please.
Thank you, Dr. Umara, for your nice words. And Assalamu alaikum to all the participants. Dr. Bano, I remember we met in 2013 in Philippines, and I'm happy to be associated with you since then. Thanks to Dr. Nasheen for suggesting my name for this event. I had some issues with my system, so I will not be able to give what exactly I have prepared for this presentation. But anyhow, I will be speaking on the PGPR. Let me share my. Can you please enable me to share my slides? To share my screen?
Say you are able now.
Yeah, so I'll be speaking on the role of Rhizobacteria in sustaining the soil health or agriculture sustainability. This is the place from where the place where I belongs to. It is 400 kilometers away from Mumbai, and this is my institute located on the bank of 2 rivers. This is my central library. So Rhizobacteria, as Dr. Humera was speaking, are the bacteria which are found in the rhizosphere. They are known by different names: plant growth promoting rhizobacteria, PGPR, yield increasing bacteria, agriculturally important microorganisms, and they were first named as PGPR by Joseph Glover. So why these bacteria or why these bioinoculants are necessary? These bioinoculants are necessary for you can say multifunction. They are responsible for controlling plant pathogens, as well as they are responsible for providing micronutrients to the crop. They solubilize various insoluble nutrients, and particularly in the tribal regions, these organisms or these bioinoculants are important for providing iron nutrition to the crops, and these iron sufficient crops can be used to combat the iron deficiency anemia. They are responsible for increasing the agricultural productivity, and they are the best solution to the chemical fertilizers and chemical pesticides. So this is the list of some of the fungal diseases of crop plants, and these are some of the pictures of groundnut infestation by Aspergillus flavus, and this is Fusarium oxysporum. These are some of the diseases of vegetable crops, fruits. So then what can be the solution to these hazardous agrochemicals? These chemicals, they are responsible, as we know that they are responsible for the development of pesticide resistance in pests. They cause addition of heavy metal ions. They destroy the useful soil flora. They are costly, they are required in large doses, and still they are less effective. Since they are chemical, they are not able to replicate themselves, so they need a repeated doses. They are not very specific to the target, and moreover, they remain in the environment for longer period. Then what's the best solution? These are some of the traditional methods, but these methods have got their own limitations. So what scientists or what soil scientists propose is the use of Rhizobacteria for plant growth promotion, as well as for biocontrol of plant pathogens. And in addition to that, these organisms can remove the heavy metal ions. So you can say their application can be the best solution to this heavy metal ion contamination. So how these PGPR and their preparations are effective and sustainable? They are safer. They do not lead to the biomagnification, as the case with the chemical fertilizers or chemical pesticides. Since they are biological, they are self-replicating, and there is no need of repeated application of this binoculant. And there are very less chances of development of resistance in the pathogen. They have multi-phase potential. They can be used as a plant growth promoter, biocontrol agent, and they can also be used for the bioremediation. They are target specific and they are broad spectrum. They are effective against wide range of fungal pathogens as well as bacterial pathogens. Since they are self-replicating, they are economical, and they can be used as a sustainable replacement to the hazardous agrochemicals. So this is how these rhizobacteria work. So three mechanisms have been proposed. One is they promote the plant growth through direct mechanisms. That includes the nitrogen fixation. They provide nitrogen to the crop plants. They produce siderophores. These siderophores are ion solubilizing compounds which are produced by PGPR, and these siderophore help in the solubilization of iron and iron nutrition of plants. In addition, these PGPR are responsible for providing a variety of minerals. They solubilize phosphate, they solubilize potash, and they solubilize many different metal ions. They are responsible for producing various types of phytohormones. So this is the direct mechanism. These are some of the indirect mechanisms. They produce antibiosis through the production of various antifungal metabolites. They create a sort of competition between the pathogen and the rhizobacteria so that the growth of pathogen will be affected. They produce various types of hydrolytic enzymes like chitinase, protease, cellulase, and these enzymes are responsible for breaking the cell wall and other components of plant pathogen. This siderophore also help in preventing the iron nutrition of pathogen so that the growth of pathogen is affected. They provide these various types of resistance mechanisms to the plant. And the third most important one is they provide, I can say, tolerance to the crop against water deficit, against the salinity, and against the heavy metal ions through various mechanisms, either through the production of abscisic acid, through the production of ACCD aminase, or through the production of exopolysaccharides. They also produce various types of antioxidants. So this is the mechanism how this Rhizobacteria PGPR help in the growth of plants. So this is what my area of research. The PGPR, they produce siderophores, and these siderophores will chelate the metal ions, particularly iron, and therefore this iron will not be available to the pathogen, and as a result, the growth of pathogen will be affected. So in our laboratory, what we work, basically we work on the biocontrol, either through the siderophores or through the hydrolytic enzymes. So we screened various soil samples, and these soil samples were subjected for the analysis of siderophore production, hydrogen cyanide, and hydrolytic enzymes like chitinase, cellulase, and proteases. So these are some of the results of screening of siderophores. These are the different isolates. Some of them are alkaligenous, some of them are pseudomonas, bacilli. And this is the medium where the siderophores are screened. This is called as chrome azurol sulfonate cas agar. So when you inoculate, initially the color of this cas agar is blue. When you inoculate the Rhizobacteria, when you inoculate the soil, and if that soil contains Rhizobacteria, siderophore producing Rhizobacteria, the siderophores will chelate the Fe from this cas agar, and as a result of loss of this Fe from the cas agar, the color of this cas will be converted into orange or maybe golden yellow, depending on the type of siderophores. So this is the indication that siderophore, the isolate is producing siderophore. And the same is the cas shuttle assay. This is the cas solution, and this is the cas solution with the supernatant of organism. So here you can see that there is a change in the color due to the chelation of iron from the medium, and this is the result of screening of hydrogen cyanide. This is the result of presence of chitinase activity, and this is the result of protease activity of the isolate, cellulase activity of the various isolates. This is the mechanism of chitinolysis, how the chitin cell wall is broken down by the chitinase. Then how the cellulose component of the cell wall is broken down by the cellulase enzyme of the Rhizobacteria. And this is the action of protease of PGPR on the protein part of cell wall component of Rhizobacteria, on the fungal pathogen. This is somewhat the new method we developed in our laboratory. Basically, siderophores are known to chelate more particularly iron from the environment or from the medium, and all the methods are available for the detection of iron specific siderophores, but there are no methods to detect the siderophores which can chelate metal ions other than iron, and we developed this method where the siderophore having affinity for copper, magnesium, and other metal ions can be screened. So then after screening the isolates for various metabolites, we went for their identification. We applied a polyphysic approach to the biolog system, GCM analysis, multi-top, and ribotyping. We identified almost more than 36 isolates, and we had deposited the sequences of these isolates in the NCBI gene bank. And then we went for the, you can say, shape flask method for the production of siderophore. So this is with the alkaligenous fecalis, and this is with the pseudomonas. So this is the medium, uninoculated medium, and after inoculation it turns into golden yellow or into the fluorescent green, depending on the type of organism and type of siderophore. Then we optimize the protocol at shape flask level, and then we also optimize this protocol for siderophore production by a statistical approach. Then we scaled up the siderophore production at 5-liter capacity bioreactor, indigenous bioreactor, as well as an imported bioreactor from Brunswick. Then we went for the purification of the siderophore on the XTD umberlite, and we purified two types of siderophore. One was hydroxylate type and another was catacolate type of siderophore. We also tested the ability or capacity of the siderophore to chelate the metal ions, and we compared their metal ion chelating ability with the chemical ion chelators like citric acid and EDTA, and we found that the siderophore preparation were superior in their capacity to chelate the metal ions compared to the chemical ion chelators like citric acid and EDTA. Then we also develop the process, optimize the process for alkaline protease production at shape flask as well as at bioreactor level. And then we tested these preparations for their antifungal potential against the common phytopathogens. So for example, Aspergillus niger, Aspergillus flavus, Fusarium oxysporum, Alternaria alternata, Cercospora arachidicola, and Metarhizidium anisophili. So we use three preparations: the purified siderophore, supernatant, and the culture broth. And we found that this culture broth preparation was more, you can say, having more potential, more antifungal potential as compared to the supernatant, cell-free supernatant, or the purified siderophore. This may be due to the role of other metabolites in inhibiting the growth of phytopathogens. So these are some of the pictures of antifungal activity of our preparations, supernatant, culture broth, and we compared these preparations with the chemical fungicides, organophosphorus, and this other commonly used fungicides. And this is the result of comparative account of antifungal activity of supernatant, culture broth, and kitazine, the chemical fungicide. This is again the antifungal activity against Cercospora arachidicola, and this is the result of our experiment of screening for hydrolytic enzymes, and their comparison with this hydrolytic enzyme rich broth, cell-free supernatant, and chemical fungicides. And again, we found that these preparations were superior in their potential to inhibit the growth of fungal pathogens compared to the chemical fungicides. So this is with the Alternaria alternata, Aspergillus flavus, Cercospora arachidicola, and then we developed the formulation for these preparations. Various types of these carriers were used, and we found that these lactose-based preparations were more effective. They were having more shelf life, and they were having, you can say, more viability. And then we went to check whether these preparations are compatible with the chemical fungicides or not. So we used three isolates, and we tested their agrocompatibility with these chemical fungicides, and we found that these preparations were comparatively stable or compatible. They can survive in presence of these fungicides. They were also compatible with the chemical fertilizers like ammonium sulfate, potash, urea, or you can say NPK. The most important criteria of, or you can say the most important factor which determine whether the inoculum or whether the added Rhizobacteria will be able to perform its functions or not depends on its ability to colonize the host plant roots. So we found that the isolates which we inoculated in the soil were capable of colonizing the host plant roots very well. And the another important criteria of any inoculum which we add in the soil is that it should not inhibit the growth of useful soil rhizobia. So we found that our these isolates were also friendly with the useful soil rhizobia like Rhizobium, Azotobacter, and other bacteria. The most and the another important criteria is the ability of the siderophore producing PGPR to utilize the siderophores produced by other bacteria, so that they will increase their potential, they will increase their capacity of chelating the metal ions as well as performing other functions. So if the inoculum is capable of chelating the heterologous siderophores, that is the siderophores produced by other rhizobacteria, it will have more potential. And this is what we tested with the medicinal plants, as well as we have tested their, you can say, bioefficacy potential with the wheat, groundnut, and other plants. And we found that these preparations were capable of promoting the seed germination, root length, shoot length, plant height, vigor index, chlorophyll content, oil content, and various other plant growth parameters. So among the 34 isolates, 6 isolates were positive for chitinase, 22 were positive for protease, and 24 isolates were positive for cellulase. Among these, Pseudomonas aeruginosa was one of the potent phytolytic rhizobia. It inhibited the growth of fungal pathogens, and it has superior antifungal activity over the chemical fungicides. It promoted the growth of plant. The another bacteria was Alkaligenus faecalis. It produced two types of siderophores. It was capable of inhibiting the growth of various fungal pathogens, as well as it promoted the growth of test plants. And these preparations, Pseudomonas as well as Alkaligenus faecalis, were compatible with the chemical fungicides. They were compatible with the chemical fertilizers. They showed better root colonization in the host plant. So since they are agrocompatible, they are biocompatible with the chemical fertilizers and chemical pesticides, they can be effective bioinoculant. They can be friendly. They are also friendly to the useful soil rhizobia. They are having good shelf life, and thus they can be used as an eco-friendly bioinoculant, or you can say best alternative to the chemical fungicides and chemical pesticides. So these are the some of the publications during 2021. And that's it. Thank you for your patient listening.
Thank you so much, Dr. Riaz, for your such a comprehensive lecture. And it is always a pleasure to listen to you. Our next speaker is Dr. Muhammad Nadeem Hassan. Dr. Nadeem, can you listen me? Dr. Nadeem?
Yes.
Okay.
Can you listen me?
Yes. Let me give some introduction of Dr. Nadeem before starting his presentation. Dr. Muhammad Nadeem Hassan is working as assistant professor in Department of Biosciences, ComStats University, Islamabad. He did his PhD in biotechnology, and his research interest includes enumeration, identification, and characterization of plant-associated rhizobacteria for biofertilizers, biopesticides, and high-value industrial products. His major research interests include the metabolic engineering of bacteria to understand the mechanistic signaling and ensure their cost-effective utilization in soil. He has developed commercialized phosphorus biofertilizer, biophos, and humiphos, which are being used as a supplements in different soils. Dr. Muhammad Nadeem Hassan has been awarded various competitive research grants by international and national funding agencies like Higher Education Commission of Pakistan, International Foundation for Science, Sweden, and Pakistan Science Foundation. Today, Dr. Nadeem will deliver his lecture on biofungicides, mode of action, and their impact on soil health. Dr. Nadeem, over to you.
Thank you, Dr. Mera, for nice introduction and rating some introductory words about me. Thank you for organizing a wonderful event, first of all, on this important occasion, that is the Soil Day. And another thing that caused the soil deterioration are the chemical fungicides. And I will discuss some suitable alternative to that, that is biofungicides, what is their mode of action and how they impact the soil. As it is evident that crop diseases is an emerging threat, and especially in context of climatic change, Their epidemic yield losses in severe with reference to epidemiological condition lead up to 100%, and with the emerging of abrupt climatic change, these threats are further aggressive, and it is a major challenge in the agriculture production. So there are various types of pathogens, including fungi, bacteria, and nematodes, which are widespread. And the emerging challenge, another in this era, is also the new races are being developed day by day. So disease area under crop production is increasing yearly at a higher rate. So what are the biofungicides? Basically, it is well aware that everyone is well aware that a lot of microbial agents have antimicrobial activity, and we can utilize these agents as biofungicidal agents. These are the naturally occurring antagonists, including bacteria, fungi, some genetically engineered, as well as a virulent mutant of certain microbes. Among these, all the agents, PGPR, termed as plant growth promoting Rhizobacteria, has a wide spectrum biofungicidal activity because they are the living microbe, they are non-pathogenic, they also improve the soil fertility by solubilizing nutrient, producing some plant growth promoting hormones, and they are cost effective.
Excuse me, Dr. Nadeem.
Yes.
Excuse me, Dr. Nadeem, we cannot see your slides. Are you not showing your screen, or?
No, I have shared my screen.
But we cannot see your slides.
I don't know. Should I new share?
Yes.
Here is everything okay. Now I have pause share. Now it is the resume share. Can you see now?
No, we cannot.
Okay, and let me share again. Now, are they visible?
No, they are not visible.
सर स्टॉप शेयरिंग करके दोबारा से शेयर कर लें. स्टॉप शेयरिंग करके सर अगेन रीशेयर करें.
Okay, I have stopped sharing, and then
Yes, now we can see. Now we can see.
okay, okay.
Carry on, Dr. Nadeem.
But now I'm not going to. I am unable to go to the next slide.
Then I really don't know what's going on.
Yes. Now.
It's okay now.
On the slide visible now?
Yes.
Okay. So why PGPR as an ideal biofungicidal agents? Because they are living microbes and they are non-pathogenic. In addition to suppressing the pathogen, they also improve soil fertility by solubilizing nutrients and producing some plant growth promoter friendly strategy. Now there is again issue. I could not go on the next slide. Okay, so while screening some potent biofungicidal agent, we start from the some in vitro tests that is include the antagonistic activity. First, we test their antagonism against various fungi on the simple plates, and based on the maximum inhibition, we do certain further biochemical and molecular analysis. They produce some important antifungal traits that includes production of hydrolytic enzyme as well as cytophore, as discussed deliberately by Professor Dr. Yasser. And then they also produce hydrogen cyanide and very important component that is antibiosis, because antibiosis are basically antifungal metabolites which are effective at the very, very low concentration. And these different type of antibiotics are further detected through LCMS as well as HPLC, some biochemical traits, as well as coupled with some genomic techniques that involve the targeting of some conserved genes which are involved in their biosynthesis. These important antibiotics, which are encoded by usually Pseudomonas and Bacillus, ubiquitously found biocontrol agents. These are the antibiotic pyrolutin, and the conserved gene is PLTP, then surfactant, this is the SFP. And these two genes are not only used just for the functional characterization of antibiotic producing Rhizobacteria, but they also act as a marker for the enumeration of these effective biocontrol agent from Rhizosphere. So then we quantify the different antifungal metabolites like protease, glucanase, and siderophores in the absence as well as presence of different pathogens. And we quantify how these pathogens act as a signaling molecule, and it also depicts their interaction. These potent antifungal metabolites are then identified by 16S or DNA technique, and they are found to be some diverse genera apart from Pseudomonas and Bacillus. Certain very, very effective metabolites have been found as a Acrobacterium intermedium and Stenotrophomonas maltophilia species. Then how they interact with the soil, because they have to perform in a complex environment, and these environmental signals and the regulation of these traits, they are called as a fungicidal traits. That is very, very important. So for this, we have characterized certain bacteria by developing different knockouts, and we have targeted the sigma-based regulation of some housekeeping genes that are very well GAC-S, GAC-A mediated regulation. Then lead-S mediate and HORP-S, HORP-L, which are also involved in the type 3 and type 4 secretion system in case of opportunistic some pathogens like Pseudomonas aeruginosa and opportunistic plant pathogen, rather fully plant pathogen that includes Pseudomonas orange. So in response to the environmental signal, there is a very, very nice regulation of these antifungal metabolites, which is dependent on these housekeeping mechanism, GAC-S, GAC-A, and lead-S, as well as HORP-L, HORP-S mechanism. Then for their in situ fitness, there are certain traits that include biofilm formation, motility. These strains have also been characterized by knocking out certain specific genes which are involved in the antibiotics, as well as the CIF or Lux type protein that encodes multiple functional traits in the Rhizobacteria. So we have also found their biofilm and motility, and then we have counted the colony-forming units in Polanta, how they have found to be analyzed, and how these Lux type regulator and housekeeping genes, they are involved in the epiphytic fitness. Then in Polanta evaluation, that includes, we have conducted so many experiments of the effective biofungicidal agent on economically important crops like rice, sugarcane, tomato, and wheat, and a significant reduction in the disease as well as increase in the agronomic traits as well as yield have been observed in pot experiment, net house experiment, as well as in the field conditions. So then their formulation development is very, very important factor, because the ultimate object is commercialization and development of the commercial formulations. So they have shown a very good survival as well as colony forming units in the different carrier materials like sugarcane filter cake, peat, and some liquid formulations also. So then academia industry linkage is very, very important. So we have evaluated these strains in collaboration with the Shakargan Sugar Research Institute and on the major crop of their interest, that is sugarcane. And we found that these Acrobacterium, Pseudomonas, and Bacillus strains have significantly reduced the disease incident up to 52, and even in some cases up to 60%. So similarly, these strains have been very well evaluated under field condition as well on the rice crop, and against blast and as well as Buchanan disease, we have found that they have again shown the efficacy up to 36 to 38%. But these strains have been isolated from the rice rhizosphere because indigenous bacteria has a greater advantage of epiphytic fitness on the specific crop. So we have also, they showed a good colonization. We have also conducted field trials across the 2 consecutive years under field conditions and in collaboration with the National Agricultural Research Center, Islamabad. And they have significantly suppressed the different varieties of the rice, Super Basmati and Basmati 385. So conclusively, these PGPR are most effective antagonistic and biocontrol agent of the crop diseases. However, their prevalence is very, very rare. We can find that most of the antagonist bacteria are found to be antagonist, but from lab to field application, there are very few agents which are ideal candidate for the development of a bioformulation. And these developed bioformulation of those very, very active bacteria, they have a potential commercial to be commercialized as a biofungicide and biopesticide. But before commercializing them, there are certain future prospects and rather challenges in the commercialization. One of them that is inconsistent performance, because as I already discussed, that these PGPR have to interact in a quite highly complex environment. So their host dependence ability to perform as well as specific pathogen interaction is very, very important. And that leads to when we cross the boundary of either it is the host or pathogen, it leads to consistent performance. Second aspect, which is usually ignored, that is risk assessment. For example, we have recently started the studies and which are under the investigation, and we will publish very soon, that these PGPR have been found to carry a lot of antimicrobial resistance genes. Hence, they could be a source of drug resistance in the Rhizobacteria. And similarly, future aspect is another, that lias like Pseudomonas aeruginosa and some other Pseudomonas strains like Ochrobactrum as well, which show the PGPR potential, but they might carry some virulence genes. So these type of risk assessment are very, very important, and every scientist working on the PGPR should focus that whether we have characterized their safe aspect, but on the other side, if we ignore that, it will cause a very harmful impact on the environment. So we must assure their risk assessment in terms of drug resistivity and carrying of the other virulence genes, and their mobility, how it can move from one organism to the other organism. So finally, I will acknowledge all the members of my team, Applied Microbiology and Biotechnology Laboratory and the research institution, that have facilitated us in conducting field trials and evaluating the bioformulation, like Shakargan Sugar Research Institute, National Agriculture Research Center, and my host institute of postdoc Imperial College London, Higher Education Commission, Pakistan Science Foundation, and International Foundation for Science, who have provided me the international and national grants and their support in this research. So thank you.
Thank you so much, Dr. Nadeem, for such a great presentation. Now,
thank you, Dr. Mera. Is there any questions? So
we can have questions at the end if someone has. First, I will invite our last speaker, Dr. Nasheen Ilyas. Let me give, Dr. Nasheen, can you hear me?
Yes, I can hear you properly.
Okay, let me give some brief introduction of Dr. Nasheen Ilyas. Dr. Nasheen Ilyas is assistant professor at Department of Botany, Pir Meher Ali Shah Arid Agriculture University, Rawalpindi, Pakistan. She did her PhD in plant sciences from Qaid-e-Azam University, Islamabad, and she is always distinct in her academic career with the first class position in masters, and did PhD programs under fellowships from Higher Education Commission, Pakistan. She is dedicated with experience in teaching and research in plant microbiology and plant physiology, thoroughly involved in teaching and supervised dissertational research work of 7 PhDs and 31 MPhil students to date, and mentoring 5 PhDs and 6 MPhil students. She has received research and publication awards from her university. She has participated in several national and international conferences and symposiums. Currently, she is the president of Pakistan chapter of Asian PGPR Society for Sustainable Agriculture. Today, Dr. Nasheen will deliver her lecture on regulation of above and below ground fluxes for combating climatic change. So I will invite Dr. Nasheen Ilyas to come and present her presentation here. Over to you, Dr. Nasheen.
Thank you, Dr. Humera. Dr. Humera has always been a nice friend and a very good colleague of mine. So I'm happy to be with her every time.
Thank you so much.
So, first of all, I am thankful to the organizers for arranging such a timely and which is the need of the time seminar or other webinar. Here we are discussing the role of the soil for particularly our hopes, our interest, and our issues with the soil on the World Soil Day. And I'm thankful to the first organizers. I'm thankful to Professor Riaz, and I'm thankful to Dr. Nadeem. They have added quite a good information to this topic. And today we are going to discuss about some of the above and below ground fluxes, which are helping in combating the climatic change. This is a continuation of what you have already heard, but some from different perspectives. So first of all, why we need to talk about this? As you have heard about the international perspective, you have also heard some importance of the PGPR, but there are some things which are related to the Pakistan, and being Pakistani, our main concern is more about what is happening in our country. There are some good things, there are some bad things. The good things, this is the general information we all know because we all are from the same country, so we can be short in it. That Pakistan is a sovereign state. We have an area of 796,095 kilometers. Important thing, agriculture. Agriculture is the main contributor to our GDP. It is contributing to 18.9% to our gross domestic production, and it is also providing a living to 42.3% of the people. And you know that we have cultivated regions, we have irrigated regions, and we are producing a number of crops. We are producing cotton, sugarcane, mangoes. We are most importantly producing the staple crops like wheat and maize. And this agriculture, this is playing a very important role in our national economy. It is providing the food to the consumer, fiber to the industry. It is earning us foreign exchange, and it is also providing us a market for the industrial goods. But the important things when we are overall having a view of our agriculture in Pakistan is that we have a great diversity. We are a diverse country, not ethnically and linguistically, but also in our geography and wildlife. Interestingly, this is the single country in the whole world which has, from the mountainous regions, from the top mountains, the highest mountains of the world, from the desert region, even we have the sea level in the same country. In the tropicals, coastals, subtropicals, all of this is adding diversity to our environment. And this diversity does means that we have harsh environmental conditions at some places also. Along with it, we have a population burst. We are among the sixth most populous country, and our country is growing at a population rate of 2.4% per annum, which is quite fast. And due to this fast growing population rate and many other issues, we have food security. Our large population, around 48% of the population is food insecure. These are some things which we need to worry about. First thing is hunger severity index. If you can look over here, we are among the countries which are threatened with the hunger severity, means we are not able to feed our population. The second is the second trend is showing the water crisis. If you can look over here, we are among the countries which are having waterless condition. It does not mean we have water shortage. It means we are totally having no water. We have waterless conditions. So this is a very critical condition, means our water requirements are difficult to be met. The third important thing, which we are all aware now, is the climatic change. You know that Pakistan is the seventh most vulnerable country to the climatic change. It means that our environmental conditions are going to worsen every year, and we are at a threat to the climatic change. And all of this is negatively impacting our agriculture. And this is where we have to focus. We have to pay attention. We have to find the alternate solutions, because it is predicted that our agriculture production might have a decline in the future due to all these environmental conditions, and these predictions are not very hopeful. So we have to feed the population, and we are having the situation where we are waterless, we have climatic vulnerability, and all of these are having negative impact on our agriculture. So we have to go for sustainable agriculture in Pakistan, and even in organic farming is also a part of sustainable agriculture. And it was estimated that only 0.1% is of the total organic farming in the world. Only 0.1% is having the organic agriculture is happening in the Pakistan, which is very less. The other important thing is soil fertility. When we are talking about the PGPR, which are the plant growth promoting rhizobacteria, which are adding the fertility to the soil, why they are very important? They are very important because our soils are very deficient. Our soil are deficient of 100% organic matter. Our soils are 100% deficient in nitrogen. We are deficient in all macronutrients. We are also having deficiency in the micronutrient. There are a number of reasons for this low soil fertility. These are sometimes inherent reasons. We have leaching, erosion, fixation, removal. All of these, they are resulting in the loss of the fertility in the soil. The other important thing is that we have low fertilizer usage efficiency. It means our crops use a large amount of fertilizer per ton production. So it means that we have to add more fertilizer to the soil for meeting our yield requirements. So these are some other fertilizer effects, and our fertilizers, as our soils are deficient in the organic carbon. So if you can compare it with the neighboring countries, this is the Pakistan, where we have the least fertilizer use efficiency as compared to our neighboring country. And the main reason is, as I have told you, our soils are deficient. So we have to add a large amount of the fertilizer to maintain or to achieve our yields. So here, telling you what is the situation here, I have tried to show some part of our work which we are doing to improve these conditions. Our work is divided into three parts. First of all, we study the stress tolerance mechanism in plants, because that is very important. Ultimately, we have to increase the performance of the plant to improve its performance, particularly under the different environmental conditions. In the second part, we are dealing with the soil microbes along with the soil amendments, because when we are talking about the soil, everything is important whatsoever is happening below the ground. Microbes are equally important, so are the soil amendments. And in the third part, we are combine both of these things together, and then we study the plant responses. This is also the part of the research theme which we carry in our lab. First of all, in the part one, we study the plant for the stress tolerance mechanism. Second part is more related to the World Soil Day and what's happening in this webinar. We first of all isolate some indigenous PGPR. Our target is mostly from the stress ecosystem. Why? Because they may help in the selection of the stress adapted strain. Then we characterize this PGPR. This is done based upon the stress tolerance, the morphology, the physiological and molecular characterization is carried out. Then we prepare some soil amendments, because there needs to be some method with which these PGPR are to be transferred into the soil. And we are working on the compost, biochar, and alternate cropping techniques. In the third part, we are combining both of these techniques, and we are studying the effect of these soil amendments and microbes, and particularly under the stress. And then we study the plant responses. We study the effect of these microbial strain and soil amendment techniques, and we study the plant, particularly for the morphological, physiological, and biochemical parameters. And in some way, we have even tried to elucidate the mechanisms which are happening under all these studies. So this is what is happening. This is what we are doing in our lab. So we have the selection for the plants that have the positive effect that leave the positive effects on the soil gases. We have microbial inoculation. We have the organic amendments. We apply the various stresses on the plant, and we study if we try to find if we find some solution. These are the PGPR. I'm not going too much in detail of the PGPR because this has been discussed in today's webinar. These are the number of the mechanisms with the help of which the PGPR are increasing or helping the plant in its productivity and growth. This is something about the soil organic amendment, how they help in improvement of the soil. Okay. This is what we are doing in our lab. We are targeting about the PGPR, we are targeting about the cropping techniques, particularly we have done some work on alternate cropping, we have compost, we have biochar, and in the end, now we are talking about bioorganic fertilizer. These are some of the stresses which have been studied in my lab. We have done work on the drought. We will focus more on the salinity because that's the theme of this year. We have worked on the heavy metal, hydrocarbon, and environmental pollution. This is what we do. This is the QTL mapping of the drought tolerance in wheat. I'll skip this drought and we'll more target about the salinity. And this is the important thing. This is the part of what we do. We go for the stressed ecosystem, we isolate the microorganisms, we go for the screening, then we screen them for the PGPR traits, and then we go for the phenotypic and genotypic characterization, and the genome which we obtain, we go for its blast analysis, and then we deposit it into some gene bank like NCBI or any other. So these are some of the very good or promising microbial candidates which are an asset of our lab. If you can look over here in the top, we have isolated from some PGPR from some plants which are growing naturally into the salt ranges of Pakistan, and they have provided us very good result. These are their accession number, and these are the amounts of APA they are producing, particularly under the salt stress condition. I hope you know that abscisic acid is the stress tolerance hormone. If some microbe has the ability to produce abscisic acid, it means that it can help the plant in combating the salt stress conditions or any other stress. Then we have isolated the microbe from the arid region of the Pakistans also. They have been isolated from the cereals, particularly the wheat and the maize. One of the important parameter which we have taken is the production of the phytohormones. These microbes, they have the ability to produce the plant growth promoting hormones, the plant hormones, and among these plant hormones, we have studied for the indole-acetic acid, gibberellic acid, cytokinin. But our criteria for this PGPR selection, it had been the stress hormone, that is the abscisic acid. Then we combine these things. We combine, for example, we have Azospirillum strain which is tolerant to the stress. Then we combine it with some cereal crop, particularly like here we have the maize, and then we study the effect of this inoculation under the stress condition. And we see for the plant parameters, we go for plant morphology, plant physiology, and biochemistry, and see that if this inoculation has helped the plant to ameliorate the stress conditions. So along with it, we also combine sometimes this PGPR inoculation with the soil amendments and study that if they are able to help ameliorate any kind of the stress in the plant. This is some of our work which has been done in relation to the drought. This is the part one, where we have the effect. We have studied the plant responses into the drought, because it's very important to know how a plant in nature is combating the stress. What are the main parameters of the plant which can provide an indication if a plant is tolerant to the stress or it's not. This is the work for our second part, where we have isolations. We have isolated a number of the strains, and these are the work where we have got them published. And this is where we have combined, and we have studied the effect on the plant, particularly after the inoculation. This is the work which we need to focus. This is where we have done for the salt tolerance. This is the QTL mapping of the 7 chromosomes of the wheat. QTL quantitative locus, you have an idea about it. Quantitative QTL, it provides an indication that what particular trait is responsible for a stress tolerance, and it is situated at what particular position in a chromosome. So these are the cuticles of the 7 chromosomes in the wheat, and this work was an extensive work where we studied for a number of the wheat germlines, and we have observed that there are some traits for it which are very important, particularly when we are talking about the salt stress. In that case, there are 2 important things. One is the osmotic stress, and the other is the ionic stress. And it's important to study the plant parameters which are responsible for these tolerances, and then we will be able to finally ameliorate the stress in the plants. This is the second part of our study for the salt region, where we have isolated the bacteria, and these bacteria were isolated from the salt stress region, and they were tested for the proline content, for reducing sugar, for the total soluble content. These are important osmolyte. If they are being produced by a bacteria, if they are produced by a bacterium, it means that the bacterium has the ability to tolerate the stress under the natural condition. In the third part, we combined this PGPR inoculation and soil amendments and under the stress condition, and we were able to find out some very good result that this plant growth promoting Rhizobacteria, then particularly in combination with the biochar, they showed the salinity amelioration, particularly in the wheat. So these are some of the publication record which we have done on the salinity. This is the publication where we have done for the QTL mapping, the rhizobacterial isolations, and the applications, particularly for the amelioration of the stresses. Along with the drought and the salinity stress, we have studied a number of the other parameters. Particularly, we have done some work on the heavy metal. We have studied the effect of the heavy metal on the plant, and we have done some isolation of some strains which have the ability to tolerate the heavy metal stress. And these are some of the whole genome sequencing of the microbes which have shown the tolerance for the heavy metal. This is also the continuation of the heavy metal. We have applied the heavy metal tolerant strain to the plant, and we have started the heavy metal tolerance of the plant after the inoculation. We have also done some work on the emerging pollutants like the hydrocarbon, and we have tested the combination of this PGPR with the biochar and studied that if they have the ability to mitigate the effect of the crude oil contamination. And these are some of the good results. We have been able to formulate a plant microbiome bioremediation system for the crude oil contamination. We have done some work on nanobiotechnology also. Most importantly, we have found a very good synergism between plant growth promoting Rhizobacteria and silicon dioxide nanoparticle, and they were able to ameliorate the drought stress in the wheat. And this is where we have now, by now we have reached where we have formulated bioorganic fertilizer. We have done some solid state fermentation with the help of plant growth promoting Rhizobacteria of the agricultural waste, and it has resulted in the production of a bioorganic fertilizer. And this fertilizer was proved to be very efficient in growth promotion, particularly of the maize plants. Okay, these are some of the work which we have. Give me a minute, please. Okay, this is where we have tested the formulation and efficacy testing, and then we can conclude this work. The system has been hanging for a minute. Give me a minute, please. Yes. Can you see the screen, please?
Yes, ma'am.
Okay. So whatsoever the work which we have done in our lab, we have concluded this work in two parts. There are two targets, particularly. We have studied some of the whole plant responses, and we have studied that particularly when we are talking about the soil stress, it is very important to have some osmo protections from the membrane stability and RO scavenging. These are very important. Until and unless we have the osmo protection, because osmotic stress is very important under the salt stress condition. Similarly, ROS are being produced under the salt stress condition, and they're affecting the membrane stability, which is affecting the homeostasis in the plant. And along with it, there are some targets which are in the shoot, and there's some targets in the root. For example, it's very important to regulate the root zone water availability and the root ethylene and oxygen levels. Along with it, it is important to regulate the stomatal regulation and the hydraulic conductance in the shoot. And these are the important parameters which we have found from our study, which can be helpful in evaluation of the stresses in the plant. These are some of the awards and honor which have been received by me and my lab. A recent one from in the field of the applied and environmental microbiology, and these are some of the other merits, awards and merit which I have received from my university. And along with it, it's an honor for me to be a part of the Asian PGPR Society for Sustainable Agriculture, and I'm the president of Pakistan chapter, as Dr. Humaira mentioned. So any one of you, if you want to join this society, we welcome you. You can contact me and Dr. Humaira, anyone who is available. We are always available for you. So we are on way for our bioorganic fertilizer and silicon nanobio fertilizer, and these are some of the nominations. And I'd like to use this opportunity to particularly thank Professor Dr. Askari Bano. She has been my mentor, she has been my PhD supervisor, and whatsoever I am today, it's all because of her. And this is, if you can look over here, this is the first picture is from my PhD, and we are together from a long time, particularly me and Dr. Humaira. So it's always a pleasure to be with her. And these are some of the students which are working in my lab. And I thank you all, and I welcome you all for your questions.
Thank you so much, Dr. Nasheen. It was a wonderful presentation, and it is really an honor for us because your achievements are really making us proud every time. And you're right that all of this is because of our mentor and our great teacher, Dr. Asghari Banu. And we are lucky that Dr. Asghari Banu is here, and she has accepted my invitation despite of her time difference, because she's in Canada, and this is her very early morning, and now I will request Dr. Asghari Banu for vote of thanks, and let me introduce her before starting the vote of thanks. Professor Dr. Asghari Banu, she is presently serving as a professor of biosciences, University of Wa, and she have obtained her PhD degree in plant physiology from University of Glasgow, UK, sponsored by Commonwealth Scholarship. She did postdoctorate from Germany, USA, and Canada under the award of Alexander von Humboldt Stiftung from Germany and Fulbright Fellowship from USA. She served Dhaka University for 2 years and Quaid-e-Azam University for about 30 years in various capacity as chairperson and dean faculty of biological sciences. She has supervised 122 MPhil and 45 PhD students and published more than 230 research papers in national and international journals, which have received about 280 impact factor with 7,840 citations. She is the author of 14 book chapters, and in recognition of her great work, she was honored with UNESCO Recitation Award of Gold Medal by Pakistan Academy of Sciences, the Research Productivity Award by PCST, and the DTS Performance Based Award. She have also supervised postdoctoral research and won 13 research projects. In 2011, she was awarded Civil Award Tamgha Imtiaz from President of Pakistan. Since 2011, she completed 6 research projects, and she have been granted 8 patents, and 2 patents are under review. So these are her achievements, and I'm feeling really proud that I am student of Dr. Asghari Banu, and now I will request Dr. Asghari Banu to come and say a vote of thanks to our honorable speakers and our attendees. Over to you, Dr. Banu.
Assalamu alaikum, Dr. Humaira. Assalamu alaikum, everyone. Thank you very much for giving me this opportunity. Thank you.
Thank you, ma'am.
Thank you. So it was a very good occasion, and I feel very privileged and honored to offer the vote of thanks to the distinguished speakers and our participants, students, and our colleague. So, honorable speakers, distinguished audience, and respected colleagues and our dear students. Assalamu alaikum again. So I have been asked by Dr. Humaira Yasmin to offer vote of thanks. Dr. Humaira is serving as assistant professor. She is a very good faculty member at ComSats University, Department of Biosciences, and she has a very brilliant academic career as well. And I have been very much pleased with 2 of my students are presenting. 2 of my students are on this very front line. So you know that, as Dr. Humaira mentioned, that this soil science, this soil day was celebrated in 2002 by the International Union of Soil Science. And since then it is going on. You know that soil is actually the hub of all the living organism, microorganism as well, and also on the macroorganism as well. So we must have to look at it. So in this context, it is our in this context, 3 of our speakers have highlighted the importance of this valuable entity, which is the foundation of our food web. It becomes a very much great achievement, which I have seen by our 3 distinguished speakers. I'm very thankful to Professor Dr. Riaz Sayyed, Head Department of Microbiology, for sharing his knowledge and expertise on the role of Rhizobacteria as health promoter for soil and plants. Soil is the hub of microorganisms as well as microorganisms, as I told, and for that matter, soil health is very important. The talk by Dr. Nasheen Ilyas on regulation of abiotic regulation of above and below ground fluxes for combating climate change is also an important, and she has very much highlighted and has a deep insight into the importance of many biotic and abiotic, particularly abiotic factors in relation to climate change. Because climate change is modulating the fluxes and also impacting the soil microbiota. So we must know about it, and I'm very much pleased to see her very much in-depth study, and I hope that that will keep it up. And I'm also grateful to Dr. Muhammad Nadeem Hassan on presenting her lecture, his lecture on biofungicide, mode of action, and impact on soil health. This is an important aspect because we must seek an alternative for eco-friendly, sustainable solution to keep the soil and our ecosystem healthy. Finally, but not least, at the least, but not the last but not the least, I am very, very grateful and thankful to Professor M.S. Reddy, who is basically the founder, chairman of Asian PGPR Society for Sustainable Agriculture with 1,500 plus members. He is basically the drive to this very important aspect in academia, and he is not only the founder, but he gives the incentive to scientists, most of the scientists, and also he has very brilliant academic career, has many awards, which I don't have time now to say, and everyone knows about him. I'm very much grateful to him that he also has given me the opportunity to participate in the conference, in the international conference in Philippines some time ago. And I always had his contact. Finally, I extend my deep sense of gratitude to Dr. Humaira Yasmin for organizing such an event, and the audience, and also the students to keep patience and make this webinar a success. Not only the students, but not only the Asian PGPR Society is very active, which gives me a boost to the academia, that also the Multi-Omics Society is also given a great weightage and deserve my great appreciation and thanks. So thank you all, and it was a pleasure not only to listen to all of you, but also it adds to my knowledge as well, and it gives the pleasure to be with you. I hope this will continue, and we will protect our soil and soil biodiversity, and also can contribute to the environmental changes and environmental changes, and will set the ecosystem to make it sustainable and purifier. Thanks a lot. Assalamu alaikum.
Walaikum Assalam. Thank you so much, dear Dr. Asghari Banu. We are really honored to have you with us in this occasion. Now I will, in the end, I would like to thank all of you for joining and for valuable time. Now I will ask Tayyab. Before that, I really wanted to thank my very dear and very talented students, Mohsin, Zafar, and Tayyab, and their team, which they are running Multi-Omics Society, and this was the effort of my students, which enable us to conduct this successful seminars. And Tayyab, can you please ask us, some participant if they have some questions? We have our speakers, so we can take some questions. Tayyab?
Thank you very much for your kind words. You have always been an inspiration for us. So, participants, the house is open for the question and answer session. We have our respected speakers with us. If you have any question, kindly type in the chat box. I will read it to them and they will answer your questions. Thank you.
I have a comment. I have a point. I hope the next time we all meet in person, and I hope the next time Dr. Humaira and her student also arrange a good cup of tea for us.
Yes, sure, Dr. Nasheen, inshallah. We have a plan, inshallah. We'll do it very
Yes,
soon.
we'll meet in person, inshallah.
Inshallah, inshallah.
House is open for the question and answer session, people. If you have any questions.
I have a question. Can everybody listen to me?
Yes, you are audible.
I have a question to Dr. Nasheen Ilyas about her second or third slide, in which she mentioned a table where potassium is deficient 40 to 50%. Can she share some reference related to that slide?
May I know who is asking the question?
The question is about the fertilizer, organic matter, nitrogen, phosphorus deficiency.
Okay.
And you have reported that the potassium deficiency in Pakistani
it's
soil
not
is...
it's not my report. I haven't tested the soils. This is what I obtained from the literature. This is not my report.
Okay,
This is
so
what this is.
So I was interested
you're
about
talking
the
about-
reference that was mentioned by that
Okay,
scientist.
okay. If you can provide me your email ID or something like that, I can provide you the reference also.
Okay, thank you. Thank you.
Any other question, please? So I think Dr. Mehra will not have much questions.
Any other question from the participants?
Ma'am, participants are just thanking us for this informative session and thanking all the respected speakers for the informative knowledge. No, there is a question, ma'am. Can I say it out loud?
Yes, you can ask. We're listening.
Okay, ma'am, there is a question from Shafia Salim that what are pesticides aptamers?
Pesticide?
Aptamers.
Aptamers. Dr. Nadeem, I think so. This falls in your expertise. Can you please answer this question?
I think pesticide aptamers are basically, this is not related with the fungicide, but as well, I think that these are some organic classification or some organic compounds which are suspended in the soil after application of the chemical pesticide.
Okay, Dr. Nadeem, thank you very much. I hope Shafia Salim, you got your answer. Let me see if there is another question. If there is no other question, then I think we can end this session. Dr. Humaira, can you please end the session?
Okay. Thank you so much, everyone. Again, I would like to thank you all, my dear speakers, Dr. Riaz, Dr. Muhammad Nadeem, Dr. Nasheen, Dr. Asghari Bano, and my team and my dear participants for having a very good gathering on the Soil Day and making this seminar successful. So I would say Allah Hafiz, and we are planning to have a series of webinars on specialization with all the abiotic stresses and biotic stresses one by one. So I hope we will see each other together very soon. So Allah Hafiz from my side, and this is all about today's seminar. Thank you all. Allah Hafiz.
Thank you. Allah Hafiz.
Thank you, ma'am, very much.
Allah Hafiz.
Thank
Allah
you.
Hafiz.
Thank you,
Allah Hafiz.
Allah
Yes,
Hafiz.
ma'am. Thank you very much, ma'am.
Thank you.
Welcome.
थैंक यू सो मच एवरीवन। हमने चैट बॉक्स में एक लिंक डाल दी है। दिस इज आवर फर्स्ट पाकिस्तान फर्स्ट रिसर्च ग्रुप। अगर आप इसको ज्वाइन करना चाहें, यहाँ पर आपको हर तरह की गाइडेंस वगैरह मिल जाएगी। थैंक यू टू ऑल आवर स्पीकर्स एंड थैंक यू एवरीवन फॉर ज्वाइनिंग अस। अल्लाह हाफिज।
Allah Hafiz.